Hello, Year 11. So we're continuing on with monohybrid inheritance. So we've already talked about a monohybrid cross and Punnett squares, and now we're focusing in on the second part of the dot point, and we're focusing in on a monohybrid test cross. Okay, so we're going to discuss what a test cross is and the success criteria. What you should be able to do at the end of the video is use a test cross to determine the genotype of an individual who's showing a dominant trait. So this is an individual who has a dominant trait. So quick recap, monohybrid inheritance and Punnett squares, right? We've got a cross happening between two flowers. We've got our purple flower, which is homozygous dominant. We've been given the genotype there. We've got our white flower, which is homozygous recessive, and we've been given that genotype there. Once we've gone and we filled in the Punnett square, we've got our F1 generation here, and these are all the possible offspring. In this instance, the only possibility is that the offspring could be heterozygous. So just a quick recap that this is a Punnett square, and we're still talking about monohybrid inheritance and Punnett. Okay, so a test cross. So sometimes we might have an individual who has the dominant phenotype, and they could have one of two genotypes. So they could be homozygous dominant, big D, or heterozygous, in this instance, big D, little d. So what we need to do is, or one of the ways that we can determine the genotype, and this is, it's still in use today. It was more of a thing in the past before we had the technology to so easily sequence genomes that we have now, but it is still currently used. So what we can do is we can perform something called a test cross. So we would cross a individual that has the dominant phenotype with another individual who we know is homozygous recessive, so an individual who has the recessive phenotype. And then we go and we look at their children and we look at their children's phenotypes and try and use that information to try and work out what the genotype is. Either is it homozygous dominant or heterozygous of the unknown individual. So a key point to point out for this process is that it sometimes will have to be done multiple times to give us accurate information, to make sure that we're doing it multiple times so that we have an accurate result, a reliable result. So in this instance, and we'll walk you through this process in a moment, but in this instance it took 7 children before we came out with this homozygous recessive child, which is actually the one that tells us that our dad must be heterozygous. But we'll break that down now. Okay, so now we're going to look at an example of a test cross, and I'm just going to turn my video off here so that you can see the entire screen. So it's the same problem we had on the last slide. So we've got a dad who's got the dominant phenotype, black fur, and we don't know what this dad's genotype is. It could be homozygous dominant or it could be heterozygous. So one of the ways that we can figure out what the dad's genotype is to perform a cross with a individual who's homozygous recessive. So this mom, we can see, is definitely homozygous recessive. They've got the recessive phenotype, gray fur, so they must be little d. So now we've got two possible outcomes, right? So on the left, if the dad is homozygous dominant, and we perform that cross here, we can see that if the dad was homozygous dominant, 100% of the children would be heterozygous. So if 100% of the children are heterozygous, that means if we were to look at their fur color, 100% of the children would have black fur, okay? So if we were to perform this cross, multiple times, and every single time we did it, the children all had black fur, we could pretty concretely say, yep, it looks like that dad has the homozygous dominant genotype. Now, if the dad is heterozygous, the other possibility, if the dad is heterozygous, it's a little bit different. We can see that 50%, So 50%, 2/4, 50% of the children are heterozygous, will have black fur, but 50% of the children will be homozygous recessive. Okay, so 50% would have the black fur and 50% would have gray fur. So what we do is basically, if we did this cross and we saw a single cat, you know, we do it 10 times, and if we saw a kitten that had gray fur, then we would know immediately the dad must be heterozygous. Okay, so we'll do another example. Okay, so let's imagine you have two flowers. We've got our homozygous recessive flower here. We know it's homozygous recessive because it's white. White is the recessive phenotype, so it must be homozygous recessive. And then we've got the dominant flower. And again, this flower could be homozygous dominant or heterozygous. Well, how do we know? One of the simplest ways is just to breed, or one of the most cost-effective ways is to cross these two flowers. And you'll have to, I'll turn my camera back on here. You'll have to excuse my daughter if you can hear her in the background. So this is the possible offspring, and it's the exact same as the cross with the cat. So if our purple flower is homozygous dominant, has two copies of the dominant allele, then it's only passing on the dominant allele. All the offspring are heterozygous, and all the offspring are showing that purple color, that purple phenotype. But if our flower is a heterozygote, we have that 50-50 split again, where half are showing the recessive phenotype because it's passed on the recessive allele, and half are heterozygotes. Okay, hopefully that's nice and clear.